This is where we left off on the last one. Remember, we're in the stomach. We are deep into the gastric pit. And this would be a generic gastric pit. I just want to start out that they would vary based on where they are in the stomach. So those that are closest to the esophageal sphincter are mostly going to have mucus. Those that are closest to the pyloric sphincter, definitely mucus, and then you're going to have more of the G cells there. We'll talk about that a little bit. But let's go down, and the gastric pits are all designed this where they have a neck feature, and then they have a little bit broader area, so it kind of looks like a bottle. And again, this is to control the fact that the contents don't want to spill out to the surface. It can spill out to the surface, but basically it's designed to keep the contents down deep in the pits. So you have the superficial or the surface mucus cells, and they secrete mucus. You have mucus cells in the neck, more mucus. You have parietal cells. Parietal cells, will show another slide about it, secretes hydrochloric acid in its own way. It doesn't secrete it as hydrochloric acid, but it secretes something that becomes hydrochloric acid. And also intrinsic factor. And intrinsic factor is important for B12 absorption. B12s, remember, are important for making red blood cells for hematopoiesis. Or I guess maybe it's not remember, you're learning that stuff. And then chief cells, so you have parietal cells, hydrochloric acid, and intrinsic factor. Chief cells are going to secrete pepsinogen and gastric lipase. So pepsinogen is an inactive form. It becomes active in the acid environment. The acid environment activates that. And then G cells are going to secrete the hormone gastrin. Gastrin tends to slow down or get the duodenal ampulla ready for the arrival of the chyme. Then we'll move on. Those are the cells of the gastric pit. We will talk about them again. One of the reasons why I use that image then is I want you to see where they kind of are in the gastric pit. So now what we want to do is we refer to this hydrochloric acid. We are looking straight at a parietal cell, and I'm on the left here. And what the parietal cell does, it doesn't actually secrete hydrochloric acid. What it does is it secretes carbonic anhydrase. That's H2CO3. And what that does is that turns into bicarbonate, saying that wrong, bicarbonate ions. So carbonic anhydrase forms carbonic acid, which when it has its charge to it, and that carbonic acid is because it's you're removing one of those hydrogen ions, and then a bicarbonate ion which has a negative charge. Then so you have a hydrogen ion and a bicarbonate ion. So their carbonic acid to provide the hydrogen ion. That hydrogen ion then is transported to the lumen of the gastric gland. At the same time, that bicarbonate ion, remember it has a negative charge, is exchanged in the interstitial fluid for chloride. So it's a negative for a negative. In terms of your chemical gradient, you have an equal there, and in terms of your electron gradient, you have an equal there. So a negative for a negative. So that chloride then moves into the gastric lumen, and that's where hydrogen chloride is made. And when that is taken back up, it goes back the other way too. But the thing of it is parietal cells, we say they give rise to hydrochloric acid, but they don't actually make it. They make the components of it, and in the gastric gland is where hydrochloric acid could do its work. But each of the pieces of the ions take different pathways in. And then the thing about hydrochloric acid is um, we talk about it helping digest proteins. It doesn't necessarily digest the protein. It provides a very acid environment again for bacteria, microorganisms. It helps denature proteins by it helps denature some of the walls, the protein walls, some of the connective tissue, and then it does this other thing about giving the environment for pepsinogen to become pepsin. Then pepsin is the protein digesting enzyme. So that's quite a bit for that to do. And then the other picture is in case that communicates more to you. It basically says the same thing, but maybe that picture works better for you. Then I like this in your book. I'm not going to read all of this to you, but I like these little places in your book where it tends to summarize all the material. And here you have it on the PowerPoint. Summary of what's happening, what cells, what they do, what is being secreted and what they do, and then also the digestive activities in terms of layers. The CNS is not relaxing in all of this, so your nervous system isn't relaxing in all this. So with this you have a couple of reflexes, and reflexes, remember, are from the CNS, but they don't synapse in the brain. These are generally involuntary, and you have a reflex that actually stimulates the motility of the stomach and secretions along the entire small intestines. And this is just a reflex action based on the presence of food. And then you have a reflex that triggers the opening of the valve. This is the ileothecal valve between the small intestines and the large intestines. So small and large, so that things can move along. So that they actually, it's a reflex coming from the stomach that dictates what's going on way away from it in terms of the tube, but allows for there to be movement out of the stomach into the small intestines. And then another great image you have in your book, the spotlights. In your book, these spotlights are good areas for you to look a little bit closer because it pulls multiple things together. And what you have is on the far left, your cephalic phase. So those things that are happening when you see, smell, and begin to taste food. So in the very beginning, you have actually a nervous system, central nervous system interaction in that cephalic phase. We talked about that a little bit already. That actually getting the stomach ready for food to come to it by getting mucus, activating the mucus, chief cells, parietal cells, the G cells, and to release their substances. So getting things ready. And then you have the gastric phase when food arrives into the stomach, and you have stretch receptors. So you've probably eaten a big meal or even not even a big meal, so it's just stretching the stomach. And chemoreceptors, so you have a change in the pH, so chemoreceptors. And then you're going to get local responses too. So you have that submucosal and a local response with regard to coming after the CNS response. You're going to have a local response to what's happening in the stomach. Getting the stomach to actually churn, etc., releasing more of those chemicals, and then you have your intestinal phase when the stomach starts to empty. So when food is moving into the small intestines, you actually have most of the chyme moving to the lower portion of the stomach, and so you have stretch receptors that are more activated in the lower portion than the upper portion. So these also, you have then stretch reflex receptors in the beginning of the small intestines, which is the duodenal ampulla, and they then are going to trigger the release of even more chemicals, secretin, GIP, and CCK, which we need to talk about. Again, you have in your book, you have the image-wise, and then also these tables, which tend to organize the material into a little bit more, little less narrative, but kind of organized, I guess, notes, I might say, that become probably after you've read the chapters and listened to the lectures, the things that you want to look at. And then control of the larger control, this kind of is similar to what we just talked about. It does tend to be written a little bit different, where you have this tends to be the last portion. This tends to be looking more at the intestinal phase, where chyme is then starting to be released into the duodenum. And you have those stretch receptors. Those stretch receptors then are going to trigger the secretion of secretin, CCK, and GIP, GIP. I'm over there on the left, and as those get secreted, that increases the contractile force of the stomach and causes the rate of the stomach emptying to decline. And then after the decline, then you're going to go back. This is a negative feedback system. Then you're going to go back to it's going to decline until you have the presence of food into the duodenum again. And then on the right, you have chemoreceptors. Those chemoreceptors are going to target your short and long reflexes. So the short reflexes for the enteric neurons, so the hormonal or the chemical communication neurons are going to be stimulated, so that the cells that can secrete the chemicals will secrete their chemicals to stimulate the local areas. And then you have, via the CNS, long reflexes, this is basically laying out the rate with which food is in the stomach. We talked about food being in the stomach about 3 to 4 hours. I guess we didn't talk about it, but it was written on there. So this actually gives you the rate with which the whole that process of the emptying of the stomach, actually all the phases of the stomach will happen. I think ultimately it's just another way to look at it if that communicates for you so much. Then on to the small intestines. Small intestines, I guarantee you somewhere along the line you're going to be asked the 3 parts. The part that's closest to the stomach is the duodenum. Middle portion's jejunum, and a portion that attaches to the large intestines is the ileum. We remember that by remembering don't jump in. So don't duodenum, jump, jejunum, in, ileum. Just be very careful about the spelling of ileum, because don't be like your teacher and misspell it all the time. Small intestines, most of the absorption takes place in the jejunum. And you do have, again, surface area. There's our famous word, the plica, to increase surface area, so that the small intestines actually has a surface area of a house that's about 2,500 square feet. Unbelievable. Large surface area. Histology of the stomach, of the small intestines. We will, well, actually, I think that probably the one that I want to refer to is the top middle, where you're actually looking at the individual. We kind of looked at this already, but what you have are the individual cells on the plica, and they secrete mucus and also other specialized cells. And within you have your blood vessels and also your lymphatic vessel. We're going to have to stop at this one and pick this one up when we start the next part.